Between our microblitz and between the forays that we did last weekend, the ones that are coming up this weekend, and between the lecture series that we did this week, I feel like we did get our fungus on this January, which is nice because there was a lot of rain and there was a lot of mushrooms out there for us to find. So I'm glad we didn't totally miss it. And I also really hope that next year, all these same people maybe have a chance of meeting up in person and seeing us at our fungus fair. But now I get to introduce Maria to you. Maria is someone I'm lucky enough to call a friend, as well as someone I work with from time to time on teaching people about mushrooms. And Maria is up in Eureka, up in Humboldt County. So I'm sure some of you have been up there to College of the Red. Well, maybe you've probably not been to College of the Redwoods, but you've been to the Humboldt Mushroom Fair. College of the Redwoods is just south of where they hold the Mushroom Fair. And I've gotten to meet Maria's students and I've gotten to see them respond to Maria's teaching style, which is totally different from mine. And she brings, I think what you're going to see is a very distinct style that I hope you enjoy. And I've learned a lot from Maria, and I look forward to working with her for years to come. And I hope that you all get to meet her sometime soon. So with that, I'm going to let Maria take over with Adventures of a Microcosmonaut.
Okay. Let's do some screen sharing and some presenting. Okay, everyone can see my screen all right? I've got some thumbs up. That sounds great. Okay, so this is a talk that I am probably not the person to do, but I really wanted it to be a talk, so I'm doing it. And hopefully someday you all with your little micro friends will go out and make your own future additions and episodes to this talk and add to it. So this is our little microcosmonaut here, Nan-O. The O is short for oh my, because this is by far the most uttered phrase of this little microcosmonaut. And today we are going to just wander through some of the little microcosms that we have been in for the past week. And I will try to identify and teach you about some of the different groups that we saw. And this will go beyond just fungi. I'll actually cover kind of a lot of plants because for some reason, the combination of super wet, but also pretty cold led to mostly bryophytes and ferns that I was running into. Also, feel free to ask me questions as I go. I'm totally okay with you jumping in. And I know that sometimes the timing can be a little bit weird where you are just going to have to interrupt me, but I will not see it that way. So if you have a question as we're going, I'm not very good at looking at the chat while I'm talking. I'll just sort of space out and stop talking, but you are welcome to jump in and ask me something. Okay, so here we go. This is Nan, and they're going to teach you about what we find in our little microcosms. So first, we're going to look at some of the duff, some of our clues for what other life is around. So you can see we're holding a cone of some kind, and this particular cone should be recognizable to everybody from this region because it's from our coast redwood. So that's Sequoia sempervirens. And you'll find these laying around in the duff. But other clues that we have coast redwood around are these feather-like branchlets. So these are different from a lot of the other conifers in that they fall as a whole group. They stay stuck together, and even as they start to decay, they just lose their color, but remain this feather-like branchlet. And they're pretty flat. So the needles don't come out all the way around. We just get these flat branchlets, and they pile up because they decay really slowly. So this creates a really interesting litter layer that can be different from a lot of other forests. So redwoods are unique in the ecosystem that they engineer. Other things that we're going to see in our duff explorations are Sitka spruce. So you can identify Sitka spruce either when it's freshly fallen by these needles that come out and go all the way around. And you can see that these pale lines here are the stomatal blooms. So these are little holes that open in the leaves and allow CO2 to come in and oxygen to come out as they photosynthesize. And sometimes if you're working on IDing conifers and you don't know the trees around you, you'll have to look at the pattern of those stomatal blooms for ID. So Sitka spruce, the needles go all the way around. They're sharp to the touch. unlike a Douglas fir. And when they fall off, they leave this little wooden peg. So you can identify spruce duff in a lot of pictures if you are on iNaturalist trying to figure out what a mushroom is and you are looking for clues. Spruce duff is really easy to recognize because of these little knobbly sticks. Okay, so let's get into some of the other plants, full plants that we'll end up seeing in this duff layer. So we might run into liverworts. Liverworts have flat sets of leaves where they come out in a single plane. And then we'll see some underleaves on the underside. But first I want to talk about this way that they reproduce. So all of these bryophytes, the stuff that Nan is immersed in right now, these are some of the earliest plants to evolve and to move on to land. So these are often restricted to wet habitats where they have to stay close to a water source. Many of them have the ability to completely dry out and then rehydrate, this poikilohydry. But we often find them in these dense mats clustered together with highly diverse assemblages. So we'll be looking at a lot of those today. So here we see those flat leaves and then this emerging structure here. Bryophytes are weird in that most plants that we see and are used to seeing with our eyes are sporophytes. So they are the part of the plant that is going to be a seed making thing. Maybe they're going to bear cones, but they are diploid like us. But here, these early plants are completely reversed. So these are all haploid, meaning they only have one set of chromosomes, and this is their tiny sporophyte. So this is the only thing that's going to be diploid on this whole plant, are these little itty bitty sporophytes, and then they're going to release spores, and those spores will grow into more of these. So notice how small this is. We just have a clear little stalk and then a single capsule at the end. If we look at the underside of a liverwort, we'll see these little things called underleaves. And that's a really great characteristic to look for to make sure that you have a liverwort and not just a flat moss. So look for those underleaves and then those very simple sporophytes. So let's see how they're different from mosses. For mosses, we have these spirally arranged leaves. They're not in whorls like we see on some trees, some other groups of plants. They come up in a really tight spiral. It makes them really distinct in their appearance, even from far away. The other unique thing about mosses and how they're different from liverworts is many of them will have this distinct midrib type structure, except it's not vascular tissue because they don't have that. It's just like a thickened part of the leaf where it can allow it to fold, but that's called a costa. So we'll see that in some of our mosses. And it's a really great, easy feature that when you're looking at these kind of green blur mats, you can look for that line on the leaf and know that you're looking at a moss. You can also look for the thing that most people associate with mosses, these more complex sporophytes. So we have this long stalk, the seta, and then we have the capsule here, and then a bunch of structures can happen at that mouth. And we'll look at some of those in later slides. This is Kindbergia. It's one of the more common mosses that you're going to run into. They just look like a bunch of feathers laying on top of each other. And then another way that you can identify a moss versus a liverwort is that the male mosses, the ones that make the sperm, will have a splash cup at the top. So this little flattened dark area is where the sperm for the moss gets produced, and then raindrops hit it, splash it out. It travels along the ground until it finds a female plant and then fertilizes the egg, and then one of those sporophytes will grow out. So a plant with a sporophyte growing out, like this one, is technically a female plant, egg carrying, and then the male plants will have these splash cups. And this is Leucolepis. These are super fun. They're like little palm trees. So here we have a mix, an assemblage of mosses and liverworts growing together. And it can be hard to figure out what is coming out of where. So we have these two sporophytes here where it looks like they're emerging from this plant. But if you look at this plant, you'll see that the leaves are sort of more flat. We have some little underleaf type structures and then no costa. So this is actually Scapania. It's a liverwort, and these sporophytes are coming out of this moss here, which I think is again a Kindbergia. So we have mosses, we have liverworts. Other things that we might run into in these dense mats of green are little ferns. So this is a young fern that has just come up and is still rolled up. So this is called a fiddlehead. And as they unravel, that's called circinate vernation. So it's unrolling, and that vernation, think of vernal, like the spring. So they start as this fiddlehead, and this protects all the growing leaf tips from being munched on by like roving banana slugs or snails that are coming through. So they stay rolled up, and as they open, those growing tips are the last thing to come out. Oh yeah, I put this up here because I knew I was going to forget. This moss here is another really common one that you'll see. They always look like kind of a little hummock, and they look like the wind has blown across them, where all of the leaves kind of face the same way. So it's called the windswept moss. Okay, so I forgot to put an animation on that one. I was going to show you that ferns on the underside will always have, at least at some part of their life cycle, will have these clusters of spore producing structures called sporangia. And we see those, that same term used when we look at fungi. So these little circles here are these deflated sporangia. And you can see each one has this dark line on it. There's a thickened band of cells that wraps around. So imagine the sporangium as a sphere, and then a thickened band of cells that wraps all the way around it, called an annulus, another word we have for fungi. So that annulus has inflated cells, and they're inflated with water. And when it starts to dry out, those inflated cells dry out and shrink, and that rips open the sporangium and releases the spores. So you can look for those structures on the underside of fern leaves, and they're often in different patterns. On this pentagramma, they're just sort of everywhere, but in some ferns, they make distinct lines or distinct dots, like on the underside of a sword fern. All those distinct little dots are called sori, little clusters of sporangia. Okay, so liverworts, mosses, ferns. What is this? We're approaching fungal territory. We start to see some lichens in here. So we have a moss in the background. You can see its little complex sporophyte there and the spirally arranged leaves. But in the foreground here, we have a lichen. And this particular lichen you'll see a lot. This is a type of cladonia, and they have complex structures. So this lichen has a few things we can look at. At the top, this bubble apothecium. An apothecium might be a term that you're familiar with if you have studied ascomycetes. So that's a whole group of fungi, the cup fungi. And these are cups, but they're turned inside out and kind of convoluted. So this is a fungal spore producing structure. So a lichen is part fungus, but it also has to have something that photosynthesizes. So in most cases, it's going to be a green alga, but we'll see a few examples where it's going to be a cyanobacterium. Usually you can tell based on the color. If they are green or gray or lighter in color, they tend to, or that tends to be your hint that there is a green algal partner. If they're more blue-gray, which we'll see in some of these, like very dark colors, sometimes black, that tends to indicate a cyanobacterium. Okay, so we have our apothecium up here, the fungal partner, and that just produces fungal spores. That spore has to land and find some green alga that's out there free living, capture it, and then start to farm it and build its own lichen thallus. Another way to reproduce is just by having all this dusty stuff. These are just little pieces of lichen that can fall somewhere and then grow into a new lichen. So the apothecium at the top can be many different colors in this group. The color of it is a great identification feature. And it sits at the top of something called a podicium. So each of these little stalks is a podicium. But not all cladonia are going to have those podicia. This one does. It's got a few podicia here, but most of it is squamules. So all of these little leafy bits are called squamules. And you might recognize that term squamulose from some of the ways that you can describe textures on fungi. Here is another lichen that you're going to see all the time. And this one has a great identification feature for it. So this is a highly branched fruticose fungus or fruticose lichen. And if you break it apart and just pull on it a little bit, you'll find this stringy, spongy cord that's got a lot of like bungee tension to it in the center. And that cord, if it's present, shows you that you have an usnea. So this is the true old man's beard. And there are tons of different types of usnea, but they all have the same central cord. And if you want to start identifying usnea, then you have to be looking at that central cord. What color is it? How thick is it? How thick is the tissue around it? So if you're going to get into lichens, you're going to have to get into this teeny stuff. You're going to want to have a little set of chemicals along with you, because just like fungi, because we have this fungal partner, we get a lot of color change in reaction to similar chemicals like KOH. Oh, there's that white cord. So this is usnea, and we'll see it again in this presentation because it's everywhere. Oh, there it is. So these lichens that have a three-dimensional shrubby structure are called fruticose lichens, but there are a few other different types that we'll learn about. So there's one back here that has a completely different structure. This one is flat. So that's called a foliose lichen, like a leaf, where it has a top side and a bottom side very distinctly. Some will just grow oppressed to a surface. Those are crustose lichens. I didn't find any in these adventures, but those don't really have a bottom. You'd have to scrape it off the surface to be able to look at it. So it kind of just has a top. So this one has a upper surface that is darker, which might indicate to us that we're looking at a cyanolichen. And if we flip it over, the underside is orange. The underside on these isn't always orange. It starts out white, but it can wear away, or sometimes it gets grazed off. This stick was flipped over, and I think potentially something had come and rasped off the lower surface of it. This is a nephroma lichen, and it's one of my favorite ones because it's pretty easy to identify. So that orange interior, and then its little apothecia, and they look like little paws. This is a very cute lichen that you should have your eye out for, nephroma. So I didn't include crustose lichens in here, but I did include dustose lichens, which are also called leprose because they fall apart into little pieces. So this you'll see on redwood trees all over the place. There's this kind of minty green one that's Lepraria, and then there's a bright golden one called Chrysothrix, and they're just dust. All of these little dusty granules contain the algal partner, and you can kind of see some of that potentially here. And then it's wrapped in the fungal partner. So they're just like little pockets of lichen that can then fall somewhere and produce more of itself. This is basically just a big pile of lichen spores. Other types of lichens, we have gelatinous lichens. These, when they get wet, turn into just goo. And often these have a cyanobacterial partner. And these have to be in pretty wet spaces for them to survive. This one's growing on a seepy bank where water's just running down it, in with a bunch of liverworts and mosses. And you can see it's translucent. So if this were to dry out, it would maybe turn to like a grayish color. But when it is wet, it's translucent in this blackish blue. It's really cool. Leptogium. Hard to spot though, really small. Kind of just looks like stranded algae. Okay, so now we're moving into this weird zone where this looks like a mushroom, and it is a mushroom, but this mushroom is part of a lichen. So not only do we have these ascomycete lichens where there's an ascomycete partner, we also have Basidiolichens. And weirdly, they don't really make a lichen-like structure. Instead, they make mushrooms, and that's where the fungus reproduces. And then this slime covering the base here is the rest of the lichen. So there is a photobiont, something that's photosynthesizing, and that's what these mushrooms are eating, sugars made by this green slime. And all of that together is one thing, or we refer to it as one thing, but there's many things involved, right? In all of these lichens, we have potentially 3+ partners. And every day people study lichens, they discover more and more things going on in there. So this slime plus these mushrooms is the Basidiolichen, Lichenomphalia umbellifera. And sometimes you'll see a log that's just covered with this green slime without anything else growing on it. And if you come back to that log at the right time, you might see these little tiny fruiting bodies coming up. Okay, so let's get into some mushrooms. We're walking around in the duff now. We're not in on a log. We're not in a crevice. We're just out in the redwood duff, these thick piles of redwood branchlets that stay together for years. Who knows how long? Somebody might know. This is the first thing that we ran into. These long, tough stipes with small kind of red caps to them. And then as these age, these caps fall off and you're left with just the stipe left. So that gives it its common name, the horsehair fungus, but its scientific name is Gymnopus androsaceus. So gymnopus means naked foot, which I think is pretty funny because never have I seen a gymnopus that has a smooth base. So I don't know where that name comes from, but I would love to find out. This is a super common fungus in redwood duff. And if you don't see it fruiting, you might see these little remnants. They look sort of like rhizomorphs, these blackened thin strands that are just sticking up out of the duff, and you can see them year round. But only every once in a while can you catch them with the caps on. And oh my, what is this? Interesting bark texture, right? This is what I do when I'm in the forest. I walk up to a tree, I look at the bark, and that helps me figure out what that tree is. But on this scale, if we were to look at tree bark, we would just sort of get lost in the crevices. It would be difficult to tell what a tree was. But we can look at the texture of fungal stipes. So for us, this fungus would just look like this. And you might see these pictures on iNaturalist frequently, frustratingly, just a top-down photo of a mushroom. But the real good identification information is in that side view and the close-up. So stipe texture gets not as much love as it deserves. Think of it like tree bark, how you can look at certain types of tree bark and identify what that tree is. If you start thinking about stipe texture in this way, you can start narrowing down your mushrooms and figure out how to ID them better. So this is a pretty old version of a Stropharia ambigua. So we have partial veil remnants here. And you can see in those partial veil remnants, we have purple spores getting caught on these little threads. So we can see that around the cap and around the stipe, because there used to be tissue that connected the cap to the stipe. Often Stropharia ambigua will have these long dangly curtains around them called marginal appendiculae. It makes it look kind of like, I don't know, a pale goth kid out in the woods. So we have a lot of purple spores being deposited. And then our last good ID feature here is this floccose stipe base. And it has been really rainy where I live. So most things that would normally be fluffy, are a bit like a wet cat. And that's what we have going on at the base of this Stropharia, where it's normally fluffy and white. And now it just looks a bit raggedy. But that would be our floccose stipe base here. And strangely, also common names. This is called the ambiguous Stropharia, and even its scientific name, Stropharia ambigua. I'd say it's one of the most unambiguous mushrooms that we have. It's one that my students can always solidly ID almost first off. So you should learn this mushroom. Okay, well, what do we have here? We've run into something completely different from what we just saw. So here we have 3 different fruiting bodies. And, I tried to pick this for Nan and hand it over, and my big, dumb human hands broke it instantly. Classic. But I wanted to show that the base is pretty solid and that the texture is really fibrous. So that fibrous texture lets us know that this isn't a jelly, because there are some jelly fungi that look kind of like this, but they don't have that same stringy texture. And this solid base is also helpful. It's like a opaque yellow. You can see it down here. So be paying attention to those details, the texture, what the stipe base looks like, and the color. So with these features together, we've got an unbranching kind of mimosa orange color, and then a yellow stipe base. So I think this is Clavulanopsis laticolor, the handsome club. And you can compare that to another typical club fungus that you can see around. This one is white, so you won't confuse it with that. But it has also a nice ID feature here, where we have a opaque spore producing area, but then this base is translucent and sterile. So the scientific name for this is Clavaria falcata. I don't know if it has a common name, but I would just call it the clear sock club because it's got these transparent shins. And here's another view of that sterile base. So this will help you identify it. The first time I ever saw this, I posted it and was like, oh my gosh, I found multiclavula, which is a club fungus that has become lichenized. And so it looks kind of like this, but it doesn't have the sterile base. And Christian, wonderful teacher that he is, turned me onto this Clavaria falcata. So I just learned about this a couple years ago, but it's all over now. It's all I can see. So we are leaving just a duff area, and we're starting to have differences in our environment that we're walking through. And part of that is because we've hit a flat spot, and in this flat spot, water collects. So water stays at the surface longer, and there's also more light. So those two things combine, make it so that plants can grow here. So we have our tiniest plants, some mosses, liverworts, and ferns, and even maybe some sedges. So here we have something that has a costa, this thickened central portion here, and spirally arranged leaves. So there's our costa. Leaves emerging in a spiral pattern. Liverwort, moss, fern. This is Plagiomnium, one of the more common forest floor mosses. There's a great book by Bruce McCune that I think just came out for identifying mosses. And it has a key in the back that's just common forest floor mosses. And you can go through that and it has, 90% of what you're going to see if you just walk around, not with a careful eye. Here's the splash cup of Plagiomnium. So I have a huge patch of male plants where it's just a sea of splash cups. I don't know where all the ladies are at, but they're hiding somewhere. Oh, the name of the moss book, I'm going to have to look up, but I think it's just Mosses of Oregon and Washington, but I'll grab it at the end of the chat because now I'm home, so I have all my books. Here's another moss that we ran into earlier, but we didn't really look at the leafy part, the gametophyte. This is another one of our most common forest floor mosses. It has a long feather-like structure where it has these side branches, and each of those side branches are all covered by little leaves. This whole structure, when you're just out in the woods, looks like one leaf, but then you zoom in closer and zoom in closer, and you see that each of these is a tiny little leaf, and each of those tiny little leaves, ooh, it's kind of blurry, has its own little costa. If you want to get into moss identification, you'll probably have to get yourself a microscope, because to identify mosses, you need to look at the leaf margin and whether it has little spikes on it and what those little spikes look like. You also have to look at the shape of the cells and whether they have thickenings where they meet or not, and a few other features for the sporophyte. So often you're counting cells, looking at cell shape. The identification is all very cellular. But for some of these more common ones, they're easier to get to. So Kindbergia is one of the feather mosses. Okay, so we also saw in that picture, I hope, that there was a mushroom. So this is a bryophytes fungus. That means bryophyte loving, and we find them growing in with bryophytes all the time. This was my most found mushroom of this week's explorations of putting this together. So we have this convex to conical cap shape. We have this translucent striate margin. So this means that at least when it's wet, it's going to have these lines around the cap margin. And what that's from is you being able to see where the gill attaches on the inside to the edge of the cap. So we're seeing through the cap a little bit, seeing where there's little gills, lamellulae, and then the regular gills where they attach to that cap. And that can be a good ID feature for certain mushrooms. So we have a translucent striate cap. and it's convex to conical. We don't have a little divot in the middle, which could lead us to a different group of mushrooms. So then we flip it over, as you always do when you're identifying mushrooms. You always flip it over. Don't worry about picking them. The rest of the fungus will be fine. You're just taking a little fruit and you're looking at it. Or you can leave it where it is, but if you would like it to be identified, you should always flip it over. So here we're looking at the underside. We can see tons of interesting features. Look at this stipe texture. We've got these long silvery strands. We have this gradient where it's fading from dark to light. We have these large cells sticking up out of the stipe, making this scurfiness up at the apex. And if we had a spore deposit here, it would look orange. One thing we can notice here, and I think I have a closer picture, but just in case I don't and I move forward, is we have really bumpy looking gill edges. Yes. Okay. So bumpy looking gill edges of this Gallerina species. And whenever you see those bumpy looking gill edges, you can kind of get the clue that if you do have a microscope and you look at this under the microscope, there's going to be some really cool cells lining that gill. And I borrowed this image from Michael Kuo, because he has lots of great microscopy photos of these large, weird cells that stick out of the gill edges of Gallerina species. So if you want to get into Gallerina ID, which I have not yet, you would also likely need a microscope and a lot of resources, because it can be hard to figure out what they are. And a lot of them right now are species complexes, where we're seeing them in groups and kind of poorly defined. Here's another one that's, I don't think a Gallerina. I think I know what this is. So this is a little redwood branchlet, and we found 2 tiny fungi fruiting out of it. And what I think these are is Atheniella arantidisca. And the reason why I think that is because we have a translucent stipe, we don't have a bulbous base, and there is this kind of creamsicle orange that fades to white. So I didn't take this one home and spore print it to make sure it wasn't a Gallerina. There are a lot of tricky Gallerinas out there, but that is a way that you could separate Gallerina out from most of the things that you might confuse it for. So Atheniella arantidisca also looks like this. So these are ones that I found that I felt more confident about, that were in a bigger group. But these are also bryophytes, and you can find them outside of the bryophyte mats, just growing in the duff as well if it's a pretty wet spot. Okay, so we're wandering more. We're back in the duff. Here we have some alders coming into our tree mix. You always want to be paying attention to what new plants are around in your overstory. Here we see more clubs. And you might look at these and think, oh, this is our Clavaria falcata. We have this white opaque top, and we have a thin, different colored stipe base. But if we look at that up close, we're going to see that it's not really translucent. Instead, it's more of like a yellow color, and it's fruiting directly out of this duff. So these are litter decayers, and I think Christian calls them forest noodles, and they fruit prolifically. So when they come up, they're in these huge patches, hundreds of them, and you don't even see them at first. You're just walking around and you see one, and then you see one more, and then you see 5 more, and then you see that you're totally surrounded in a forest of little noodles. So these are, unlike Clavaria falcata, which will usually be coming out of the soil, emerging directly from the duff. That's not growing through that plant, it's just coming right out of it because that's what it's eating. These are Macrotyphula juncea, the slender club. Okay, so we're combing around in the duff and we come on a half-buried log. Half-buried logs are excellent finds. This particular one had something that I find very rarely. So if you look right where Nan's hand is here, you see what my husband called little butt cheeks. Normally in botany, we describe these as heart-shaped, but they could also be butt-shaped. So we have a thin little flap of tissue, and on the bottom, you can see on this one, on the very bottom, there are little strands sticking out of it. These are little root-like structures, but they're not true roots. They just help it grab onto a surface. So this heart-like structure. So when we were looking at mosses and liverworts, we were seeing these leafy plants, and those were the gametophytes. Ferns are a later evolutionary group. They come onto the scene and have to compete with bryophytes and liverworts, which have started to colonize most of the available spaces that would be good for growing on. So ferns have to get taller, and they rely less on this gametophyte stage and instead invest more in their sporophyte. So this is a fern gametophyte. And I almost never find these. The sperm and eggs are produced usually on the same one, so they're usually hermaphroditic, not always. And the sperm will be released, and then they'll get washed through and then find an egg from another one as they travel through the water underneath them. So bryophytes and ferns are highly reliant on water to be able to disperse. And you can see here we have a tiny little moss growing in there, not a liverwort because it has a costa. That's all you need to know it's not a liverwort. So here's our little gametophyte. And then we can see some early emerging sporophytes. Notice the difference that these gametophytes are more translucent and the sporophytes are more opaque. These have more protection to them. They are intended to be longer lasting. They are going to go out into the world, and this gametophyte is going to kind of stay down here and then just wither away. But the sporophyte is going to feed off of it and then become a big fern frond, or a little fern frond, because this is as big as that fern's going to get. And I don't actually know what fern this is because I never learned ferns. So if you know and you want to say in the chat, I would appreciate that. Okay, we're going to climb out of the duff, up a tree. And the first thing we come in contact with is a mushroom I don't know. I don't know if this is a hemimycena or what. I don't think so because the top is dark. And I don't know if hemimycenas have dark tops. A lot of the little white mushrooms growing out of wood, just everybody will call them a hemimycena. I don't know if that's true. I get the feeling it's not, and that they're just maybe poorly understood and poorly described. And that somebody out there is doing some really hard work to figure it out, I hope, and that maybe someday we'll benefit from that. But this is one that I never got around to figuring out what it was. I did look a little bit, but unknown. It does, however, have decurrent gills, which is a good feature for later if we were to try to find out what it was. So we're going to keep climbing up this tree. And then we run into an even tinier white fungus. And again, a lot of people would just put this up as a hemimycena. But something I learned from Christian is that hemimycenas will never have this little swollen base. So we'll zoom in on this in a little bit. But this tiny little white fungus here has a swollen section around where it meets the tree. And hemimycenas won't have that. You can see it's a little bit hairy, fuzzy looking. So the thing that I think this is not Mycena ascendens. So that's the frosty bonnet, but I'm not sure. I could be totally wrong here. But I was reading about Mycena clavularis, and it is supposed to have a larger base. And I haven't seen Mycena ascendens have this like kind of stripy looking base before. Normally I've seen it have a smaller base, less kind of ornamented and distinct. And it's supposed to have more of a granular top. So on the outside, as opposed to being more fuzzy, it would be more granular. However, super tiny, and neither one have I studied in great detail. Clavularis is also supposed to smell bleachy, and I did not end up getting up close enough to get a whiff of these because they're deep in this little crack here. Maybe Nan smelled them, but you know, face mask, who could say? So once we get to the top of the tree, we're going to stop. We're going to have a little drink of water. We're going to look up at the canopy. We're going to see something really cool. So here we see another bryophyte, and we can notice a few things about it. You could notice that it has these little dark dots, which you could probably guess what they are if you're a little bit familiar with bryophytes. And then the pattern on the leaves, if you look, they look almost like stained glass or corrugated like greenhouse material. They're all wavy. So this is a great ID feature that will lead you right to the genus for this one. So these have large leaves, and they have these sporophytes that are tucked in as opposed to exerted. So having this little tucked in sporophyte and these large wavy leaves, we can identify this as Neckera menziesii. It has a terrible common name. I think it's Menziesii's Metaneckera moss. So if you are in the game of coming up with common names, this is a beautiful, beautiful plant and could go for a much better name. Then we climb out on a branch and we reach for this one, but our mask is open. And this has a very strong bleachy odor. So often mycenas will have bleachy smells, but they're so small that people aren't usually holding them close to their face to look at. They're just looking at them from above saying, a little white mushroom. So I encourage you to smell every mushroom. In particular, smell these mycenas, because sometimes they'll have like an iodine type odor or a bleachy odor, and that can help you identify them. This one, I'm not sure what it is. It's growing out of wood and it has a bleachy odor. So it might be Mycena abramsii, but that one tends to be darker. They can fade as they age, but even these little ones down here were not that dark. So I'm not sure on the ID, except for that it's a Mycena. And then we tumbled out of that tree, but landed next to something pretty interesting. So here we have a little evidence of something that I was looking for on all of our excursions and I never found. These are the leavings of a banana slug, which you tend to find just kind of draped across by some other kind of centipede type thing that I didn't know what it was. Okay, so we were doing a lot of climbing, a lot of jumping around. It got a little intense. So we just decided to stop and sit at this log, because that's the real way to experience these little microcosms, is to just take time and sit and look around. Because stuff starts to emerge, like this whole green slime around us has texture. It's these little bubbles of green algae. This could be part of the Umbellularia california, no, that's a tree, Lichenomphalia umbellifera. This could be part of that thallus, or it could just be some free living green algae forming these little colonies. We also see more mosses. And we can zoom in on those mosses and see some complex structures that they have to their sporangia. So here we have these lines that are coming out, and these are little teeth that interact with moisture in the air and can reach in and kind of pull out spores or close this capsule, depending on whether it's moist out or not. So they can interact with the environment to help in spore dispersal. So we have a stalk here, the seta. We have this capsule, and then the peristome. And that means around mouth. So the teeth are around the mouth. So the peristome. And if you get into Moss ID, you'll have to be looking at peristomes and counting cells and layers in the teeth. We can also see something that we saw before, but in a different form. So this is more redwood foliage, but it's got these weird structures on the ends. So if you have looked at trees, you have likely seen pollen cones. You've probably seen big swaths of pollen coming out in the spring. This is where redwoods make their pollen. So redwoods still make spores, just like mosses and ferns, but they make the spores inside these little sporangia. And then those spores grow into little pollen grains still inside the sporangium. Then they get released. And that pollen grain is the entire male gametophyte. It went from being this big leafy version to just a tiny little 4-celled pollen grain. that does its whole life cycle inside this little sporangium before it gets released, and then goes and tries to find a female cone and an egg to fertilize. We also see another lichen. So this is a foliose lichen with a distinct top and bottom. And we can find some spore producing structures here too. So we have apothecia, and these ones line the edges and they tend to curl around. This is one of the pelt lichens, Peltigera. And for these, you always want to look at the top and at the bottom, just like a mushroom, because you might have a different color here. You might have little pockets, and the color and branching of these structures, these root-like structures called rhizines, will be important for identification. Because this one has a smooth upper surface and it doesn't have any vegetative propagules present, no little lobules falling off, no little powdery granules, it's all just smooth. So it's not asexually reproducing. So that brings us to Peltigera neopolydactyla, new many finger. And we find another Galerina. And here we can see the spore deposit. If we zoom in, we get these orange spores that are catching on these large cells at the top of the stipe. There are a lot of Galerina species in our little microcosms of bryophytes. And another Cladonia, but this one's a bit different. We have potentially an upper surface that's been eaten away or just forms this way, but it's almost completely transparent, and you can see the algal layer here. So this is all the green algae that lives inside this Cladonia thallus and feeds it. And it's primarily just squamules, these little leafy bits. And probably the coolest thing to me that I found was this little hairy snail shell. So not many snail shells that I've seen have been hairy. However, apparently many snails as juveniles will have hairy shells that they then lose. And as I was reading about hairy snails, they only live in humid climates, which is kind of the opposite of plants. Plants that have really densely hairy leaves are often in really hot, dry environments, capturing the moisture, not letting it escape. But hairy snails, theoretically, need these hairs on the outside of their shells to be able to stick better onto the vegetation and have kind of more surface area and the stickiness of water that helps hold them onto those bryophyte mats. So these will crawl across the bryophytes, eating bacteria, eating plant cells, and just sort of grazing on whatever they can find. And I waited for a while for it to come out, but it decided not to. So I think this is Vespericola columbianus or columbianus, but it could be a juvenile of another species. But this texture and pattern on the shell looked a lot like images of this Vespericola, which means evening living. And then if we were to look up from there, we would see that there's just a canopy of spider webs. And this is something in the orb weaver family, I think, but I couldn't figure out what it was. I didn't get a clear enough picture of it. But I did get many of them on my face as I walked around. So we're back in, we're crawling over logs, we're looking at sticks on the ground. And one of the most common things that you'll see are these little yellowish orange discs. They don't have much of a stalk to them. And the top is pretty flat. They're small, they're sort of featureless. This is likely a Calycina citrina. It could be a similar species, but microscopy is required to figure out and differentiate between them. But this is a commonly identified species. Whether it's as common as we think it is, I don't know. But it's spread and you're likely to run into it. It used to be called Bisporella. Okay, definitely gumdrops for Lego people. So here's a really blurry picture, but here's the thing I was trying to look at. So this is a crust fungus, crustose. It's resupinate, grows fully oppressed to the surface. And luckily for us, this is one that actually has some features. Often they are flat or just kind of bumpy, and that can make them really hard to identify. You generally need to do microscopy and maybe some chemical tests to figure out what your crusts are if you're going to get into crust fungi. So this one has teeth, and the teeth are sort of flattened and irregular. I didn't come to a solid ID on this, but the two things that seem to stand out the most for their flattened and coarse teeth were Cristinia or Xylodon. I put some KOH on it and nothing happened. So not super informative. But then we found another toothed crust. So this started out just on the edges, bumpy, but you can see teeth forming here. And if we were to look further down where it's more developed, we'd have these more distinct teeth. And for this one, we have a feature. And I hope the volume's not on this, but yeah, okay, cool. So I'm going to put some KOH on it. And you can see it instantly and vibrantly turns this purple color. And that is a reaction we get actually in a lot of lichens, is either a yellow or a purple KOH. We also get that on a lot of fungi. And I always thought that it was a change in pH, since KOH is a very basic solution. However, when I was reading about a different fungus having a KOH reaction, it said that the KOH dissolved a purple pigment in it and brought it out. So I'm really curious as to why the KOH reactions that we do see happen. Oop. I guess we're watching it again. So that one was Mycoacia uda. And that is a crust that I run into every once in a while. It's either surprisingly common or I've just learned to keep my eyes out for it. But it just looks like a yellow flat sheet. You get up close and it's got those tiny teeth and it has that vibrant purple KOH reaction. Here's some more purple stuff. We have a little flat jelly that's forming this little spatula. And then behind it, there's some kind of melted waterlogged jelly behind it. But if you have a purple jelly coming out of wood and it has that spatulate structure, you can be fairly certain that it's Ascocorn sarcoides. And that spatulate structure is its asexual state. So that's when it's just reproducing itself, not trying to mate with anything and switch up its genes. And this gene or this species is the one that's known to have that asexual state associated with it. So it'll also make little purple cups. If the asexual state is not present, it's kind of hard to tell what species it is. So it's pretty helpful when it's around. And then on the other side of this log, I found this tiny little shell-shaped mushroom and totally thought it was going to be Resupinatus. I got pretty excited because I've never found Resupinatus. But if you see here, it's got a little stipe to it. So then there were some more on the underside, all looking like these beautiful little shells just stuck to the underside of the wood. But each of them had a little stipe. So not Resupinatus, but I'm not sure what it was. This could be a Hemimycena. It's not a genus I know well. But there was one super beautiful one just with glistening drops in the sunlight. And it had this really distinct color around the stipe. So if anybody does know what that one is, I would love to know. But my searches brought me nowhere. And then back on the ground, we ran into a little bit of poop. And this poop is particularly interesting. I always find myself running into poop. And we have some fungus growing out of it. So it's unclear to me whether this fungus was growing out of the hair in the poop. So it could be something that eats keratin, or whether it's growing out of plant matter or something else. I'm not very good at identifying poop yet, but there was a tiny little Phycomyces starting to grow out of it. So this is something in an earlier evolutionary group of fungi and a pretty well-studied group. So these produce a bright yellow beta-carotene pigment. And because they produce pigment, people thought early on that they were closely related to algae. So that's where it got that name Phycomyces. It's one of our earlier named fungi. And this is probably a little whisker. So whatever made this poop is eating little hairy whiskered things. And I really want to know what this white fuzzy stuff is, because I think I've also found it on an owl pellet. So I think it eats keratin. And then we run into some more weird looking stuff, just weird jellies on a stick. And this is another ascomycete. So again, instead of making that cup-like structure, the cup is inverted and wrapped around, and it's making spores over this outside surface. And this is called the red leg jelly belly, which I really love because this base can be a lot more purple or red, particularly when it hasn't rained quite so much. Everything was a little washed out for us. But I run into this one quite a bit. Cudonia cercinans. And just a sea of Mycena gallopus. So this is one of my favorite Mycenas because it has a good ID feature. So gallopus means milk foot. And if we break off the stem at the base, we can get some white latex. And this is from one of these mature specimens. But I found one that was really young, sorry, little young fungus, and I broke it, and we got a lot more latex coming out of it. So it can be pretty scant in older specimens, but in a younger specimen, it tends to be pretty milky. And so this is something you should also test with your Mycenas if you're trying to ID them, is to break them at the base and kind of just milk them and see if anything comes out. These tend to fruit in pretty big troops below redwood, so they're pretty common, and they don't have a bleachy smell. Here's another one that has an interesting oozing reaction. So we have a purple color, it's growing out of wood. Often these will grow in little clusters, but this one was by itself. And if we break off the base, we get this dark purple fluid coming out of it. And we can see that on Nan's hand here. It looks kind of like blackberry jam. Or blood, which is how it got its name, Mycena hematopus, the bloodfoot. Okay, so I think I'm getting pretty close to time. So here's a coral. It's got flattened ends. Those flattened ends help you identify it as Tremelodendropsis tuberosa. We have other corals that don't have flattened ends, but instead have these opaque little spikes, and they are really finely pointed and completely white. So this completely white one is actually the same as this not completely white one, but one has been infected by an ascomycete called Helminthosphaeria. And often these ones that have been infected turn a darker color and get misidentified as Clavulina cinerea, which is kind of a purple-gray version of this Clavulina coralloides. There it is, all infected. Gotta love infected stuff. Okay, last little bit here. So how do you find out what something is when it's weird and tiny and new? I can only do it sometimes. So whatever this thing up here is, I never figured out. I didn't try very hard, but this one down here was really intriguing to me because it had a lot more features. So we looked at this one up close and we saw that it was all fuzzy around the edges and that it was an ascomycete because it had that cup-like structure. It has a little stipe, you can see here, a short stipe, and the internal surface where it's producing its spores, the fertile surface, is yellow. So we came to Neodasycypha, which was fun because then I looked it up on iNaturalist and Christian had one of the first observations that sort of confirmed the appearance for me. So that was great. So how did we figure it out? We went to our bookshelf and we pulled out the Fungi of Temperate Europe. Normally, see here on the top, Mushrooms of the Redwood Coast is my go-to book. But if I'm looking for something weird and tiny that I'm not normally going to run into, I'm going to pull out volume 2 of Fungi of Temperate Europe. This is where you look for your crusts, your ascomycetes, your weird clubs that are tiny and strangely shaped. And then you get these big wheels. So this is a totally different identification structure than many books have, and I love it. So each one of these little pizza slices has pictures of things in that group, has drawings of features that you can look for, and then has some short description. So when I was looking at this, immediately was drawn to this hairy little disc over here. And I was looking at those descriptions and it really sounded like it fit what I was looking for. So then I get to flip to that section and I come to another wheel. And again, I'm looking for what seems like what I saw. I looked up close at this one, but eventually found myself drawn to this one over here because here we had a dark surface and here we had an orange surface. So we find our way to another wheel where we look for all these hairy ones with light colored surfaces, and we get to Neodasycypha. We get a look at that page. And on that page, it gives us an explanation about it. And this is the one that has a purple pigment that dissolves in KOH. And so we tested it out and we did get a very faint purple KOH reaction on some of these where it was deposited, but not all of them reacted. I'm not sure if that's because it was too dry or if I don't have the right ID, but I think I do. I also don't know what all these little dark globules are. Okay, I really wanted to show you a bunch of slime molds because I wanted to talk about their biology, but this is literally the only one I found. And I don't know what it is because when they're still in this plasmodial slime state, they're really hard to identify, sometimes impossible. So if you are interested in slime molds, you should check out the following Instagrams. Marin mushrooms, so good. Allison is amazing, and she takes pictures of the tiniest stuff. This one right here reminds me of a mushroom from the Fantasia animated series, just dancing around. Very evocative of really fun stuff. Stewcumber, so good at macro photography. Berry web images takes macro photography of a diversity of things. Here is that frosted bonnet. So our Mycena ascendans when it's first starting to emerge. And then wild macro, berry web images, who takes, nope, yep, maybe. That's berry web images. Wild macro is Tim Boomer, who takes pictures of a lot of galls too, which I really like. So all of these people have amazing macro photography, and Instagram is a great place to see it. If you don't use Instagram, I don't know what to tell you, except for I'm so proud of you for resisting. Keep up the good work. Does anybody have any questions?
Maria, that was so cool.
Thanks.
Thank you.
Somebody asked if I used focus stacking, and I did not. I have a little clip-on 15x lens that I just put over my cellular device and take pictures with it.
I am so impressed. Those photos were like amazing. They were so, I felt like I was tiny, which is basically a wish I've had for a long time.
Me too, except for all of the scary stuff about being tiny. And yeah, those little clip-on lenses are really cheap, easy to use. I would highly encourage people to get them. There's a few good brands out there. The one I have is pretty clunky and it covers the light filter on my phone. So it always makes it look like it's really dark out. But there's a smaller brand that I can't remember, but I have the Zenovo one. Oh, right. I'm also going to show you my books. Okay, so if you are interested in lichens, this one is really good for crust lichens. It's like the only good resource that I have found for them. So this is Microlichens of the Pacific Northwest, and I can send out a list of this if people want, but all the good stuff is by Bruce McCune. Macrolichens is for the leafy ones, the fruticose, the foliose, anything that's not crustose, you can find in here. Also by Bruce McCune. Common mosses of Western Oregon and Washington, also by Bruce McCune, so good. This has just like great keys, beautiful pictures, and it's little, which is nice. Because then when you're looking at your fungi books, this is the Fungi of Temperate Europe set. It's so good. You should definitely get it if you are interested in expanding your ID skills. The wheels are wonderful. Rarely do I find a weird thing that I can't sort of approximate using those books. So I would highly encourage you to get those if you're interested in expanding your sort of ID portfolio, especially for little and weird stuff. And for slime molds, there's a bunch of out of print things that I really want to get. So I have Sarah Lloyd's books, which are also really good, but they're based out of Tasmania.
Yes, and if anyone's been busy scribbling down the names of those books, the talk tonight will be on our YouTube channel that you can find on our website. And in a day or so, I'll probably have a direct link up to the talk also.
I can also put them in the chat if that gets saved.
Are those little brown gallerinas poisonous?
I don't know. I just assume that all gallerinas could be poisonous. And they don't really have like an edibility value because they're so tiny, depending on your perception of edibility value. So I've not actually looked into it. I don't know. But I wouldn't experiment with it either. But I'm kind of a coward about those things.
Thank you.
Checking the comments on the YouTube chat. Looks mostly like you have oohs and ahs. And thank yous.
I did a little bit of underlog looking today, and I was wondering if you have like a taxonomy of states of decay of logs, because I found that there's like a big difference between a brand new log and then an old log that's so decayed that there's almost less under the oldest stages. And there's this sweet spot right in the middle where there's like chunks that are forming cavities that like things like to curl up under, little salamanders and things. And then it also provides a little bit more breathing room for the mixos, it seems like. Mm-hmm. And basically I found myself wanting like a way to talk to myself about what kind of log I was looking at, like a categorization scheme for, I don't know, all the different relevant variables of logs and little worlds. Have you found anything like that or developed anything like that yourself?
I have not developed anything like that. However, there are studies of wood decay succession. So successional communities that will move in at different stages of decay. I'm not sure how much they apply to just like wood out in like laying out on the ground, or whether it's looking more at like wood for commercial value. But I have seen a handful of studies. So it's not just like somebody did one study. I think it's something that people have definitely dug into, is that wood decay succession. And those would be my search terms.
Got it. Lori asks, do you find any entomopathogens in mosses or on rotten logs?
You can find some, and I don't know, would cordyceps be considered entomopathogens?
Could be, yeah.
Yeah, so there are fungi that attack insects, and you can definitely find those, because there are, like we found one in, or a group of them in Seawood Cape Preserve here in a rotten log, because there's a lot of insects in there and it's a great place for fungi to live. So there were wood-boring beetles that had laid their eggs in there that had developed into larvae. And those grubs had gotten infected by a cordyceps fungus that then grew up out of the log and just looked like a weird little club fungus sticking up out of the wood. So I'm sure there's much more than that, but it would be, if there weren't a lot of entomopathogens in that environment, I'd be surprised because of the amount of insects and the ripe conditions for fungi. And my favorite resource for scat ID is actually, I just take a picture of it and send it to my colleague, Karen Rice.
There's also specialists on iNaturalist who are like level 4 or level 5 trackers and scatters.
Yeah, we have a local Humboldt person, Kim Cabrera, who's on iNaturalist as bear tracker, and they are incredible at IDing things. So you can post scat to iNaturalist, and people can use that to identify it. So that's one of, like you can post bird sounds, many things that aren't a picture of the actual organism, but that are good enough features that allow for ID. And then there's one in here. Are there many of those fake little mushrooms like the Lichenomphalia species out there? Lichenomphalia is fairly common. And I am guessing that we will find out that a lot of our little like hygroscopy species, maybe even some Galerina species, who knows, things that are typically bryophilous or found in those environments are potentially parasitic on those bryophytes, or that they're lichenized and we just don't know it. I can't say for sure, but I think that's probably likely, particularly with things in the Hygropheraceae, the waxy cap family. That's my guess. And I don't see multiclavula very often. It's like little white clubs that stick up out of a much lighter green covering than the Lichenomphalia. It's more like the green on the wall behind me, and then little white clubs that stick out of it that kind of yellow, but much more rarely sighted. And I think those are maybe the only two Basidiolichens that we know of here, but then there are far more in the tropics, but only, it's like less than 1% of all lichens are Basidiolichens. So it's pretty rare in general. I can also make a Google Drive folder of all of the pictures that I took, because I took like 1 million. And if anybody wants to use them for anything, they can just be open use. There's just a lot of little Lego figure, awkwardly balanced, looking at a thing. And I have no idea why Mycena smell like bleach. These are questions that I definitely have. This is part of why, like me as the person giving this talk, I want somebody else to give it so I can learn all of the things. But I definitely wonder why Mycena smell like bleach. Where is that purple pigment coming from? I have all of these questions, and I hope that more people go into fungal chemistry and find out for us. But I imagine it could be something that's like an insect deterrent, would be my best guess, based on nothing.
Okay, I was just looking to see if any of the chemists present wanted to chime in on where that bleachy smell came from, but two good questions. Where does it come from, and why?
I think my most wanted underlog tiny worlds organism to see is the one that Allison Pollock posted, that is like a little yellow slime mold, but it's actually a bacterium, the myxobacteria.
Oh,
If anyone is online naturalist, go look at myxobacterium. It's totally bizarre, and if you see it, be sure to upload it and share with us where you found it.
It's so tiny too. I think Allison has scale photos of the end of a pencil or a little probe or something that show just how tiny it is. You would really have to be on the lookout for it, because otherwise it would look like maybe a little hint of orange. But it's also really cool because bacteria aren't supposed to be multicellular. So that's one of those structures where it's like a bunch of unicellular things collaborating to make a multicellular structure and blurring the lines of multicellularity. Nice.
Okay. Question from Brennan. So do you want to unmute Brennan and ask yourself? Okay.
You can also read it if you're shy.
Now, can you talk a bit more about the relation between fungi and lichen? Are they in different families, or are they both part of the fungi kingdom? Well, go ahead. You can.
Okay, so fungi, super diverse group of organisms. Within that group, we have multiple other groups, and some of those different branches have become lichenized, meaning that they have obligate relationships with other organisms to make a body structure and eat. So fungi need sugar to eat and to live, and they have to get that from some other source. They can't make it themselves. So in a lichenized relationship, that fungus is farming some photosynthetic organism. Most often it is a green algae. So there's little unicellular green algae living on wet surfaces everywhere. Sometimes you'll see big kind of tufts of orange fluff on trees that are pretty damp. And that is Trentipolia. That is one of the green algae that typically gets swept up into this lichenized relationship. So it's a fungus and an algae, or a bacteria or both. And when the fungus is associated with a cyanobacterium, that cyanobacterium can also fix nitrogen. So not only is it getting photosynthesis potentially from that bacterium, but it's also getting nitrogen. So it's farming these unicellular organisms, completely controlling when they get to reproduce, how they grow. If lichens are in areas that are too wet, that never dry out, the fungus will respire the lichen to death. So there has to be some balance in that relationship where there's dry time, where the fungus has to focus on maintaining that body and keeping everything together and patching up holes and protecting it from sun damage. And then that's when the photosynthetic partner gets to eat. So always a fungus, at least one. And there's one fungus that forms the main body structure. There's a thin layer of the photosynthetic partner, usually a green alga, sometimes a cyanobacterium. And the cyanobacteria, if they're in like a 3-part relationship, tend to be in these little globules on the top. However, now that people are really looking into lichens and that we can do better microscopy and that we can do sort of genetic sweeps and figure out what DNA is inside an organism, we're finding that there's yeast, so little unicellular basidiomycetes inside many of our lichens. Who knows what they're doing, but they're causing some physical change. Maybe it's a different color, a slightly different shape. They're having some result on the physiology. Others are living in there, these dark septate ascomycetes, that we don't know what they're doing, but they're also inside plant cells, they're inside other fungi, they're just inside a lot of stuff doing mysterious work. We're also finding that the fungal partner that's building that body is secreting a lot of exudates that stick the lichen thallus together, and that there are yeasts and bacteria living in that extracellular matrix, potentially helping hold that body together or feeding on those exudates. So it is a vast, diverse community that's very like meta, like inside fungal hyphae, there are also bacteria. And so inside any fungus, you're going to have a whole ecosystem. Inside any organism, there's just layers of diversity. So we used to think at least 2 organisms. Now we're like, how many are in there? I hope that answered your question.
He says, thank you. So I guess it did. And just on an unrelated topic, let's see, unless there are any other questions. Our virtual fungus fair is continuing for a couple of more days, and I was just looking at the Mycoblitz component, and I see, Christian, we just went over 500 species.
Wow, cool. And I've barely done any reviewing yet, so maybe I'll be able to fish a few more out of there. And if you've already posted stuff and you're waiting for someone to look at it, I apologize. I know Alan Rockefeller has been doing a lot, so thanks to Alan for that. But I will go through eventually and help.
Yeah, and some people are a little remiss, like I am. Most of my pictures are still on my camera. We'll get downloaded and added over the next week or 2.
That's what happened for our Mycoblitz, is that we had a lot come in that window, and then they trickled in afterwards as people uploaded them. Because it's just by the date that you take the picture, not necessarily by the date that you upload the picture.
Mm-hmm.
Oh, Allison's here. I'm sorry I didn't find any slime molds, Allison. I was out there, I was turning over the leaves, I was looking at the stuff, I was trying to follow the guru guidelines, and I got nothing. I don't know if I just didn't have my right eyeballs on or if I just got unlucky. But someday I will find some slimes and I will talk to people about them. But in the meantime, you can go to Allison's talk whenever that comes up.
Okay, well, if folks are done asking questions and they're not going to unmute themselves and ask or leave questions in the chat, it's 8:50, getting close to 9. We can let Maria go. Maria, someone is asking, do you have an Instagram?
Yeah, I'm on there and I'm also on iNatural says this, Yip-Kaye. But I put most of my fungal related posts on HB Mycological Society. So you can follow me on either of those. I've started doing a mycology term of the day, which has now become a mycology term of the every couple days, where I try to post pictures of different features and get people in on the terms. And if you follow my Yip-Kaye one, you'll see a lot of like donations accepted at this place. GoFundMe for this person. So if you're not interested in that, then don't follow me.
The mycology terms that have been coming up on the HB Mycological Society Instagram have been an amazing learning resource. I encourage folks who are just starting out to definitely follow that and use it to sort of guide your learning.
Nice, thanks.
All right. Good night, everyone. Good night, Maria. Thank you so much for that awesome walkthrough. Tiny little, we should start naming state parks that are only 1 meter wide.
Oh, I would love that.
Yeah.
And then that would make surveying way easier and more interesting.
Yep. Okay, good night, everyone. Thank you to Maria for being our last speaker of the virtual fungus fair. See you all next year in the flesh.
Thank you for having me. You're all wonderful.
Bye.
Right.
Thank you. Good night.